Hi everyone, welcome to the final lecture of MD 6028. So again, this is going to be a mini lecture. Lecture 12 and lecture 13 are going to be covered in the tutorial that's coming up. The topic is going to be the same. The topic is going to be the way in which we can use viruses therapeutically in research and also in industry. So the different ways we can actually utilize viruses to our benefit. So it's actually a really important part of virology that we need to understand. And this is what we're going to cover today. So in the previous lecture, we focused mainly on bacteriophages. So we discussed the different ways that we can utilize bacteriophages, and most of that revolved around using the antimicrobial properties of bacteriophages and also utilizing the ability of viruses to be of bacteriophages to be used as vectors, so as a means to deliver genetic material into a host cell. That's what we covered in the last lecture, and what we're going to cover today is how we can use some of the other viruses in medicine, research and industry. And a lot of the discussion today is going to be around lentiviruses. Now, as I'm sure you know, lentiviruses are retroviruses, and they therefore integrate their genome into the genome of any cell they infect. This is a defining feature of lentiviruses, a defining feature of any retrovirus. And this is the central feature that we tend to utilize when we're using lentiviruses in medicine, research and industry. We're using their ability to integrate DNA into cells. As we've talked about before, it can be quite difficult to introduce new genetic material into cells. Cells aren't very good at taking up genetic material. They're not designed to do that. So as we said in the last lecture, one way we can get around that problem is using bacteriophages, but another way is using these lentiviruses, because again, lentiviruses have this natural ability to come into a cell and integrate their own genome into the cell genome. So that's very, very useful for us, again, from a therapeutic perspective and a research and industrial perspective. So that is what we're going to talk about today. First of all, we want to define this idea of genetic manipulation, because this is actually what we're talking about. We're talking about genetic manipulation. We're talking about taking target cells and changing the genetic makeup of these cells. So fundamentally, that is genetic manipulation. Specifically, what we're usually talking about is introducing new genes into the cells. However, as you'll see as we progress through the lecture, that's not all we're talking about. We're also talking about the ability to knock down genes, and that can seem counterintuitive, how we can use lentiviruses to actually knock down genes, so essentially to suppress genes within the target cells, but we can do it. It's a little bit complicated, but we can do it, and we do it, and it's quite an important technique that's used particularly in research. Just a very quick reminder that this is the process we're talking about. We're talking about lentiviruses being able to come into a cell, so to infect a cell, and as part of this infectious process, to integrate their own genome into the cellular genome. Again, if we remember, this integration is a central part of the infectious cycle of these lentiviruses. If this isn't clear, if this doesn't sound familiar, you need to go back to the lectures when we were actually talking about these retroviruses, because again, lentiviruses are retroviruses. Lentivirus is a classification within the retrovirus group. So really, we could be talking about any retroviruses when we're talking about the ability to introduce genetic material into cells. It's just we tend to use lentiviruses because lentiviruses are better at it. They're the retroviruses that are most efficient at integrating their genome into the host cell. So that's why when we're talking about these vectors today, when we're talking about utilizing viruses to induce genetic manipulation, we're generally talking about lentiviruses. But really, you can replace the word lentivirus with retrovirus most of the time, because lentiviruses are retroviruses. So hopefully it's clear that these lentiviruses have the ability to integrate their genome into the host cell. So they have the ability to change the genetic makeup of the host cell. So how is this useful to us? How is this useful therapeutically and in research and in industry? Well, what we can do is just in a laboratory environment, we can add genes to the lentiviral genome. And that means when the lentivirus infects a cell, it not only integrates its own genome, but it also integrates the genes we've added. So if we want a cell to start expressing gene X, we just add gene X to the lentiviral genome, then we challenge the cell with the lentivirus. Then if infection occurs and then integration will occur, the cell is then expressing the gene X that we added to the lentiviral genome. So a really, really powerful method that we can use to force cells to express a specific gene. There's a little bit of a complication, however, because I keep using this term infection, but that's really problematic because one of the ways we use this strategy is in gene therapies. So we want to take patient cells and we want to make these cells express a gene, so a healthy gene that is going to overcome a pathology. And the way we do that, again, we just add the gene to a lentiviral genome, then we allow infection of the cells to occur, then integration of the lentiviral genome along with the gene we've added into the host cell genome occurs, and the cell is then expressing the gene we wanted it to express, and hopefully the protein that's produced is going to counteract the pathology we wanted to target. That's the principle. The problem, however, is in that scenario, we have taken patient cells and we have infected those cells with a lentivirus, and usually the lentivirus that's used is HIV. And obviously that's not a viable strategy. It's a non-starter because we can't introduce those cells back into the patient because they have been infected with HIV. So how do we get around this? The way we get around it is we make what we call pseudo viruses. So the lentivirus that we introduce the gene into and that we then infect the cells with, it isn't a full lentivirus. It is what we call a pseudovirus. And what we're going to look at in the next slides is exactly what we mean by this term pseudo. Pseudo, as I'm sure you know, means false. So pseudovirus, it's a false virus. It has the properties of a lentivirus, and again, the key property that it has is the ability to integrate its own genome into the host cell genome. However, it doesn't have all of the properties of a virus, and the properties it doesn't have is the ability to spread infection and to infect new cells within the host. And that's what we're going to talk about in the next slides, because it's actually really, really important. So what we want to do now is think about this pseudovirus. So think about what this pseudovirus is, and think about how we make one. So fundamentally, we want a pseudovirus, so a virus particle that is capable of infecting the target cells, so is capable of introducing its genome into the target cells and then integrating the genome into the target cells, but then is not capable of producing new virus particles. Does that make sense? Because we want the integration in these target cells, but we definitely don't want these target cells to actually be producing new virus particles. So we don't want full infection. We want to really avoid that. That's the only way these strategies can be viable, is if we're 100% sure we're not having an actual infection event, so we're not having new virus particles being produced. We're just having something that looks like infection of these primary cells. So we're just introducing the virus particles, and then we're seeing integration of the viral genome into the host cell genome. And this is how we make a pseudovirus, which again is a virus that is capable of doing that. It's capable of entering into these target cells and integrating its genome, but it's not capable of producing new virus particles from within this target cell. So let's look at this slide here. The first thing that's done is the viral genome is split into 3 parts. It's split into plasmids, which you'll note are circular kind of DNA constructs. So each of these DNA constructs encodes the viral genome essentially, or encodes a third of the viral genome. So we don't need to know really what encodes what, although you can probably figure it out from the names, but we don't really need to know that anyway. What we do need to know is that between these three plasmids, the entire viral genome is present, and using these three plasmids, an infectious virus can be produced. The transfer plasmid on the left, this is one we want to pay particular attention to, because this transfer plasmid is where we keep the gene of interest. So the gene that we eventually want to introduce into a cell, we insert it into this transfer plasmid next to a third of the viral genome. So we have the viral genome split into three, included as three plasmids, so a third of the genome in each plasmid, and in this transfer plasmid we have our gene of interest. Okay, next we can actually make the pseudovirus, and the way we do that is we take these three plasmids, we just put them into cells, and we just do that in the lab. Now, putting plasmids into cells in the lab is pretty easy. These plasmids don't stay forever. Eventually the cells will fundamentally spit them out, eject them from the cell, or degrade them. But what you can do is put these three plasmids into the cell for long enough for the three plasmids to be expressed. Now, as we've said, between these three plasmids, they contain all the genes required to produce new virus particles. Okay, so they contain all the genes required to produce new virus particles that are then capable of infecting a cell. And that's what happens. So you put these three plasmids inside cells, these cells start expressing the plasmids, and new virus particles are made, and these virus particles are pumped out of the cell. I've done all this, by the way. This is something I've done in the lab, and you're literally just you're forcing the cells to take up the plasmids, you're waiting for them to start expressing them, and then you're collecting the media off the top of the cells, because the media is just packed full of virus particles, because these viruses are being made by the cells. Crucially, and this is really crucial, only the transfer plasmid is included in these new virus particles. So these new virus particles are being produced, and they are exactly the same as normal virus particles belonging to the lentivirus family. However, they don't have a full genome. They only have a third of a normal genome. They only have the transfer plasmid, right? And the transfer plasmid only contains a third of the genome. It's really important we remember this. So these virus particles that are being produced, these are actually considered pseudovirus particles because they only have a third of the normal genome that the virus would have. They also have the gene of interest, right? Because we added our gene of interest into this transfer plasmid. So in each of these new virus particles, the transfer plasmid is present, and within that plasmid, we have the gene of interest. And these are the virus particles that we're collecting, and these are fundamentally referred to as pseudoviruses or pseudovirus particles because they have a third of a genome. Now, because they have 2/3 of their genome missing, that means when we take these pseudovirus particles and we do exactly what we talked about before, we throw them at different cells, we let them infect the cells, and then in doing so, they're introducing their own genome along with the gene of interest into the cell's genome. Again, this process is called integration. This can happen, that's absolutely fine, but again, because the pseudoviruses only have a third of the genome, they're not able to produce new infectious particles. They need the other 2/3 of the genome to produce infectious particles. And this is why we use pseudoviruses, because this is the situation we want, right? We want to be able to take patient cells, we want to be able to introduce pseudovirus particles into these cells, and these pseudovirus particles will then integrate their genome along with the gene of interest into the cell's genome. but then they can't make new virus particles. So we don't have full infection. We don't have real infection. What we have is the pseudovirus genome being integrated into the cell genome along with the gene of interest. It's quite a complicated process. Again, it is a complicated thing. We're talking about level 6, but it's really, really important. This is done in a lot of labs. This is a really well established way that we can introduce genes into cells, and it's really important that we understand how we do it safely, because it is used in gene therapies. And what we're going to look at in a few slides is the way in which some specific gene therapies use this strategy. So the key takeaway here is that we're forcing the target cells to express a new gene, a gene that we've chosen. And the way we're doing that is using pseudoparticles, or pseudoviruses, or pseudovirus particles. And the reason we're using these pseudovirus particles is because we don't want the target cell to start producing HIV. We don't want actual infection. We just want something that looks like infection. And again, hopefully I've sufficiently explained how we make these pseudovirus particles. It does all logically make sense. It can just be a lot of information to take on at once. So if it doesn't feel completely clear, I'd encourage you to go back, listen to what I've said again. If it still doesn't make sense, there are resources online that should complement what I'm saying. But again, just give it a bit of time and I think it will make sense. Okay, and again, we have the process here just summarized as a bit of a kind of a flowchart. So modify viral genome in plasmids. So we take the viral genome, we split it into 3 plasmids, and in the transfer plasmid, we add our gene of interest. Then we add these 3 plasmids into cells. Then once these plasmids have been expressed, these cells will start producing pseudovirus particles. When these pseudovirus particles are created, we collect those pseudovirus particles, and we remember these are pseudovirus particles because they only contain 1/3 of the viral genome, and as part of this 1/3 of the genome, they also contain our gene of interest that we added. Then we take these pseudovirus particles that we've created and we infect some target cells. And again, we're saying infect because it's not real infection, it just looks like infection. We have the first stages of infection where the virus particles or pseudovirus particles will enter into the cell and then integrate their genome, and the genome is expressed. However, we don't have the later stages of infection when new virus particles are made. Okay, so that's the overall process that we're looking at. And again, what we're talking about is using lentiviruses to introduce genetic material into cells. Sometimes you'll see the phrase lentiviral vector used. Exactly the same thing. We're using the lentiviral genome as a vector, so we sometimes refer to that as a lentiviral vector. Now we can think a little bit about what genes are we actually introducing into these target cells. And really, the sky's the limit. We can introduce whatever genes we want, whatever genes we're interested in. I've been using the example of gene therapies where we want to introduce genes that counteract a pathology. Absolutely fine. That's definitely something that's done. But when we're doing lab research, we can just do any protein. Well, we can do a gene that encodes any protein we're interested in, really. And I've got a few examples here. This is an example of some research I did a few years ago where I used this technique to actually knock down a protein. Now, as I'm sure you know, some genes will encode things like siRNAs and shRNA, which is similar to siRNA, and an siRNA is a strand of RNA that will knock down RNA within the cell. And what you're essentially doing is you're suppressing a gene by knocking down its RNA. So you can actually use these lentiviral vectors to knock down proteins in a cell. And the way you do that is you force the lentiviral vector to introduce an siRNA. So it's the gene of interest is a gene that encodes siRNA that is knocking down the protein you want to knock down. And again, this is what I've done here. So on the left, this is a Western blot, the first figure, and you can see in the right cell line there's these thick black bands, so the protein is there. In the left cell line, there are no bands, so the protein and the proteinase reef is not there. And if we challenge both these cell lines with HIV, this is just normal HIV now, what we see is the cell line on the left, where I've knocked down the protein, is about 300 times more susceptible to HIV infection. So what I was essentially doing in this experiment, I was knocking down a specific protein to show that this protein protects the cell against HIV, and I proved that by showing that when I knock the protein down, infection goes up 300-fold. So this is a little bit complicated, but I still wanted to show you it because it's one of the real ways that we apply this technique in the lab. But as with the example I've listed on the left, there's really no limit to what we can do with this technique in terms of if there is a gene that encodes a protein and we want to express it, we usually can. We just add it into the lentiviral vector and then we follow the process. Okay, and those are the fundamental principles underlying the technique, really. What we want to look at now is gene therapies. So again, we're applying those principles we just talked about. We're applying the technique specifically to gene therapies. So firstly, what are gene therapies? Gene therapies are just any therapy that involves genetic manipulation of cells. Fundamentally, that's what it comes down to, gene therapy, right? And we're using genetic approaches to manipulate cells in a therapeutic manner. So the ultimate goal is to have therapeutic benefit for patients. Now, often gene therapies will be used to counteract disorders that have a genetic component. Right? Because if you have a disorder or a pathology that's caused by a specific mutated gene, often causing that gene to not be expressed or to be dysfunctional when expressed, so it produces a dysfunctional protein. One way we can deal with that situation therapeutically is to introduce the healthy form of the gene, so introduce the wild type form of the gene, and the idea is that by doing that we can alleviate pathology, and that's how a lot of gene therapies work. So the process, the principle, the idea is fairly straightforward. The way it's done can obviously be quite technical and often involves technical techniques like the one we've talked about earlier in the lecture. Some other therapies that we can class as gene therapies, what I just described is what we traditionally think of as gene therapies, but there are other techniques that involve genetic manipulation of cells therapeutically that we can kind of lump together with gene therapies as well, one of which is CAR T cell therapy. Which is what we're going to talk about. Well, one of the things we're going to talk about today. So I'll jump right into that now. So CAR T cell therapy, essentially it involves modifying an individual's immune cells, again genetically modifying them in a way that enables these cells to attack cancer cells. So one of the issues with cancer is that cancer cells are they're an individual's own cells, so it's very difficult to target these cells without also targeting other cells, healthy cells. That's why chemotherapy has such severe side effects often, and the reason is because the drugs used are highly toxic. They will attack any cells that are replicating fast. Cells that tend to replicate fast are those that are producing hair, so follicles, the lining of the stomach. So that's why we get vomiting and hair loss and things like that associated with chemotherapy. And the fundamental issue is that the drugs used are targeting self cells. The idea of CAR T cell therapy is, it enables modification of immune cells so that these immune cells will target cancer cells. It's an interesting idea. It does work, and it actually is used now in a number of countries, including the UK. The actual process itself involves taking immune cells either from the individual who needs to be treated or from another individual, and then using lentiviral vectors, which we talked about earlier, to introduce new genes into these cells, and these new genes will encode what we call chimeric antigen receptors. These are receptors that the modified immune cells will then be producing because they're expressing the gene. Once they're expressing these chimeric antigen receptors, they will have an affinity for cancer cells. So these chimeric antigen receptors allow the immune cells to interact with and target the cancerous cells. So the immune cells are being modified in such a way using lentiviral vectors that they are then able to attack cancer cells. Really interesting technique. Again, it is used, it does work, so it is something we should be aware of. And hopefully we can see at the center of this technique is the use of these lentiviral vectors. So the use of these vectors to genetically manipulate the immune cells. And again, just giving you some papers here to demonstrate that it is used and it does work. Okay, now what we have here is a more traditional example of gene therapy in the sense that a malfunctioning, a dysfunctional gene or a mutated gene encoding a dysfunctional protein is being replaced by a healthy copy of the gene. So again, what we traditionally think of as gene therapies. The pathology we're talking about here is retinal dystrophy. Retinal dystrophy is a pathology that occurs when the visual cycle is disrupted by an inability of RPE65 protein encoded by the RPE65 gene. If this protein is not functional, then retinal dystrophy can occur. And we can see on the right here this diagram that shows what we call the visual cycle. Now this visual cycle, it's the visual cycle, it's how we respond to light, it's how we see things, and it has to be a cycle because there is a reaction that is, as we can see again here, that is mediated by exposure to light. So light mediates this reaction. It mediates 11C retinal into all trans retinal. Retinal, and then RP65 is essentially reverting this. So it's undoing that, it's replenishing the cycle, it's allowing the cycle to continue. Obviously, if RP65 is not functional, then there's a problem here. The cycle can't continue, and this is what occurs in cases of retinal dystrophy. Again, a very good candidate for gene therapy because I hope you can see that in such an instance of retinal dystrophy, if the faulty RP65 gene is replaced by or overridden by a healthy RP65 gene, a wild type RP65 gene, then that should be a good way to alleviate the pathology because it allows the visual cycle to continue. And this is exactly the case. This is exactly what's happened in these cases. The development of a gene therapy that aims to specifically do that. It's not amazing, it doesn't work all the time, but it has some success, and there's published papers showing that it does have some success. So you'll often see the name Lux-Turner used to refer to this this therapeutic approach. Lux-Turner is a brand name, really. We're talking about Virology. It's just a way to introduce the healthy form of RPE65 into an individual with the pathology. Now, the way it's introduced is different to what we've talked about so far. So we've been talking about lentiviral vectors. A different vector here is used, and that vector is called adeno-associated viral vector. Now, adeno-associated virus, often shortened to AAV, is a virus that works in a similar way to some retroviruses in the sense that its genetic material stays present in the cell. So adeno-associated virus, when it infects a cell, it will have circularized copies of its genome inside the nucleus. It's not the same thing as permanent integration, but these stay in the cell for a long period of time. So it's seen as a viable vector to use for gene therapies, and it's used in Lux-Turner essentially, and the principles are similar. This gene of interest is introduced into the viral genome, so the RPE65 gene is introduced into the AAV genome, and then that produced complex is used in the gene therapy fundamentally. It's a long process to get to that point, but that's the fundamental, and it enables this gene of interest to be expressed in the target cells. And again, once RP65 is expressed RP65, the protein, is produced and the pathology is alleviated. Now you'll notice that this is a very recent paper. You'll also notice that we're talking about very specific examples. That's because we have to talk about very specific examples because very, very few of these approaches have been approved. The ones we're talking about now are the only ones I'm aware of. That I may have missed one or two, but we're really talking about very, very low numbers. This Lux-Turner CAR T cell therapy, and we're going to talk about another one in a second. Not to say these are the only gene therapies that have been approved, but these are the only ones using these viral vectors that we're talking about here. So it's very interesting, it's very new, developing area. So yeah, it's really worthy of our attention. Okay, what we have here is the use of gene therapies to treat SMA. So SMA is spinal muscular atrophy. Again, it's caused by a genetic mutation. It's caused by a genetic mutation in SMN1. SMN1 is essential for motor neuron survival. So if you don't have functional SMN1, then you can trend towards the atrophy of these neurons, which is defined as muscular atrophy. Again, spinal muscular atrophy. So quite a severe pathology. And the principle is, again, if we can replace this faulty gene, if we can replace this defective gene with a healthy gene, with a wild type gene, then hopefully we can alleviate pathology. And that's fundamentally what's being done in this study here. It is interesting, it is promising, it's still fairly early days, but it is interesting. The vector here, by the way, is the adeno-associated virus vector again. So the same principle as the example we were just looking at. It's the adeno-associated virus. It doesn't fully integrate its genome into the host cell. However, it does, as part of its life cycle, it maintains circularized copies of its genome in the nucleus, and these circularized copies can be expressed for a long period of time, and that period of time is therapeutically useful. Okay, and here we have just evidence that the FDA has approved this particular strategy. So Zolgensma, it sounds strange, Zolgensma, it's like Lux-Turner. You can tell their brand names. They've essentially been made up to sound like scientific names. So we want to be quite aware of that. It's okay to use the brand name sometimes, but as long as we're using it as a brand name, what we really care about is the actual name of the intervention being used. The only exception is when there's only one brand available. So if we have one company that's developed a technique and they're right at the forefront and that's what's being used, that's the only one being used, then in those cases we will sometimes use the brand name kind of synonymously with the actual name. That's absolutely fine, but we just need to be aware that sometimes when we see two names for an intervention, one name is the brand name, the other name is the technical name that we care about. Obviously sometimes the technical name is quite difficult, quite complicated. That's another reason we can lean towards the brand name. Okay, so those are some gene therapies. Those are the 3 gene therapies that are using viral vectors that have been approved, that we care about, that we need to know about. Again, some of them are in their early days in terms of usage and being rolled out, but the fact that they're approved shows that there is evidence there that they work, that they're safe, and that's why we care about them. That's why we're interested in them. Finally, what we want to talk about are the oncolytic viruses. So again, we focused a lot on gene therapies today, which is exactly what we wanted to do, but we also need to be aware that there can be some other uses of viruses, because that's the overall topic of this part of the module, isn't it? How can we use viruses for our benefit? And as the name suggests, oncolytic viruses are viruses that will target and kill cancer cells. Onco, cancerous. Lytic, we know lysis is the destruction of a cell, right? So just like when we were talking about oncogenesis and we were talking about oncogenic viruses are viruses that are implicated in the creation of cancer, genesis, and onco meaning cancer, genesis meaning creation. Oncolytic viruses, on the other hand, are viruses that are implicated in the lysis of cancerous cells. So when we're talking about these oncolytic viruses, we're often talking about viruses that have been modified in some way. So they've been genetically modified themselves in such a way that they gain the ability to target and destroy cancer cells. This is a good paper on the right here, which is essentially giving an idea about these oncolytic viruses, and encourage you to read it. Here they're defined as a novel drug class. Maybe, I guess it is a kind of drug class. Better to define it as a therapeutic intervention, though, maybe. But the principle is the same. They're used to target cancer cells, and often they're genetically modified viruses, and through this genetic modification they've gained the ability to target cancer cells. Not always, but generally when you read about this approach, that's what's being discussed. Again, this is a very recent thing. If you look at the papers, 2018, this is a bit of an emerging technology. It's been discussed for a while, but we're seeing it take off a little bit now. And as we often see with emerging therapeutic approaches and emerging technologies, it can be a little bit difficult to define it as a whole because it's still emerging. This is a good example, however, if we want an example of an oncolytic virus that is being used therapeutically. It's Imlygic. Again, that's the brand name. We have the technical name as well. It's essentially being used to target melanomas. It's again a genetically modified virus that has been attributed. It's gained the ability to specifically target cancer cells. So if you want an example of an oncolytic virus, this is a great example. You'll be able to find other examples at various stages, but this is a good one. Okay, thank you everyone. We'll call it a day there. I hope we all found it useful. This is the last lecture of the module, so thanks for listening to all these lectures. Thanks very much for engaging. It's great. I hope you found them useful, and I'll see you at the final tutorial. Thank you.